This video is on genes. So let's start with kind of some basic definition. A gene is the basic physical and functional unit of heredity. Genes, which are made up of DNA, act as instructions to make molecules called proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. The Human Genome Project has estimated that humans have between 20 and 25,000 genes. Every person has 2 copies of each gene, one inherited from each parent. Most genes are the same in all people, but a small number of genes, less than 1% of the total, are slightly different between people. Alleles are forms of the same gene with small differences in their sequence of DNA bases. These small differences contribute to each person's unique physical features. In the Operon model of prokaryotic gene regulation, groups of genes coding for related proteins are arranged in units known as operons. An operon consists of an operator, promoter, regulator, and structural genes. The regulator gene codes for a repressor protein that binds to the operator, obstructing the promoter, thus transcription, of the structural genes. The regulator does not have to be adjacent to other genes in the operon. If the repressor protein is removed, transcription may occur. So to go in a little more detail, in genetics, an operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. The genes are transcribed together into a messenger RNA strand and either translated together in the cytoplasm or undergo trans-splicing to create monocistronic messenger RNAs that are translated separately, such as several strands of messenger RNA that each encode a single gene product. The result of this is that the genes containing the operon are either expressed together or not at all. Several genes must be co-transcribed to define an operon. Transcription is the process of making an RNA copy of a gene sequence. This copy, called a messenger RNA molecule, leaves the cell nucleus and enters the cytoplasm, where it directs the synthesis of the protein which it encodes. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype, which means that we have an observable trait. The genetic code stored in DNA is interpreted by gene expression, and the properties of the expression give rise to the organism's phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism's shape or that act as enzymes catalyzing specific metabolic pathways characterizing the organism. Regulation of gene expression is thus critical to an organism's development. All right, so we have SRY in here. This is a quick one. Testes determining factor, which is TDF, also known as sex determining region Y, which is SRY protein, is a DNA binding protein, also known as gene regulatory protein transcription factor, encoded by the SRY gene that is responsible for the initiation of male sex determination in humans. SRY is an intronless sex determining gene of the Y chromosome in therians, which is placental mammals and marsupials. Mutations in this gene lead to a range of sex-related disorders with varying effects on an individual's phenotype and genotype. A human artificial chromosome, which is HAC, is a microchromosome that can act as a new chromosome in a population of human cells. That is, instead of 46 chromosomes, the cell could have 47th, with the 47th being very small, roughly 6 to 10 megabases in size instead of 50 to 250 megabases for natural chromosomes, and able to carry new genes introduced by the human researchers. Ideally, researchers could integrate different genes that perform a variety of functions, including disease defense. Alternative methods of creating transgenes, such as utilizing yeast artificial chromosomes and bacterial artificial chromosomes, lead to unpredictable problems. The genetic material introduced by these vectors not only leads to different expression levels, but the inserts also disrupt the original genome. Human artificial chromosomes differ in this regard, as they are entirely separate chromosomes. This separation from existing genetic material assumes that no insertional mutants would arise. The stability and accuracy makes human artificial chromosomes preferable to other methods such as the viral vectors, yeast artificial chromosomes, and bacterial artificial chromosomes. Human artificial chromosomes allow for delivery of more DNA, including promoters and copy number variation, than is possible with viral vectors. Yeast artificial chromosomes and bacterial artificial chromosomes were created before human artificial chromosomes, which first appeared in 1997. Human artificial chromosomes are useful in expression studies as gene transfer vectors, as a tool for elucidating human chromosome function, and as a method for actively annotating the human genome. All right, and then we have gene cloning. Gene cloning is a commonly used molecular biological technique in which a gene of interest is fused into a self-replicating genetic element called a plasmid, which when introduced into a suitable host, which is usually bacteria, self-replicates and generates a large number of identical copies of the particular gene. In a regular molecular cloning experiment, the DNA containing the desired gene is isolated from the organism. This DNA is cut into the right size using restriction enzymes. The plasmid, which is used as the vector, is also treated using the same restriction enzymes. Cutting with the same restriction enzymes ensures that the ends of the vector fragment are compatible with the ends of the DNA fragment that needs to be cloned. Next, the DNA fragment and the vector are mixed together and allowed to ligate together. Ligation is performed using ligase enzymes. The recombinant plasmid, which now contains the DNA fragment as an insert, is transferred into the bacteria through a process called transformation. The bacteria are then grown on growth media containing specific antibiotics that allow only plasmid-containing bacteria to grow. Bacteria are grown in large numbers, and plasmids are isolated from them. Each of these plasmids would carry the gene of interest as the insert, which if needed can be isolated using the same restriction enzyme that was initially used to insert them into the vector. Gene cloning allows for making large numbers of copies of the desired gene in a matter of a few hours. The cloned gene can be used for DNA sequencing studies, for the production of large quantities of a desired protein by cloning into an expression vector, or even to cure disease using gene therapy. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve the body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases, such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Gene cloning is also widely used in the field of agriculture to create new strains of crops and new and improved varieties of various fruits and vegetables. Gene cloning has been used to transfer nitrogen-fixing genes of bacteria into food crops to boost food production without using expensive fertilizers. In the pharmaceutical field, bacteria containing genetically cloned plasmids are used for the overproduction of vitamins, hormones, and antibiotics.